Good morning, guys. Well, it's probably afternoon now, but it's still morning where I am, or when I'm doing this. So today we start a brand new topic. We're done with topic 2. Now we're moving into like the juicy good stuff. We're talking about genes. So 3.1 is all about genes and chromosomes and that heritability. So how you get those traits from your parents. Understanding number 1, a gene is a heritable factor that consists of a length of DNA and influences a specific characteristic. Now, the word heritable means you can inherit it from a parent, right? Remember, there's no length associated with the gene. It's however many nucleotide base pairs it takes to create the protein that you're trying to create. So, like we discussed in topic 2, DNA is the molecule of life that determines the nature and characteristics of cells and organisms. And it's stored in the form of chromosomes in the nucleus of eukaryotes. Now, that's a little misleading. DNA is not stored in chromosomes all the time. Chromosomes only form when the cell is getting ready to go through mitosis or meiosis. Most of the time, your DNA hangs out in long, loose strings called chromatin inside the nucleus. But for the sake of conversation, today we're just going to think about DNA as being coiled up in these nice packaged chromosomes. A single gene is a section of DNA that determines or influences a trait. They are heritable, which means they can be passed from parent to offspring. So here we have our DNA molecule. Here we have the double helix. We have that sugar phosphate backbone. We have those nucleotide base pairs that follow complementary base pairing rules. Now, through the process of transcription and translation, we end up with a protein. And depending on if there are mutations in these base pairs, right, if there are, this protein can be either defective or malformed, which can result in differing traits. Now, this example that we're looking at is sickle cell anemia, and we're going to talk a lot about that in this particular subtopic. Here we have a normal red blood cell, and here we have a sickle red blood cell. But it's important to remember that not all mutations are bad, right? Mutations are the reason why you see so many different types of life on this planet. Without them, we'd all be the same. Actually, we'd all still be bacteria, but that's another story. Understanding number 2. A gene occupies a specific position on a chromosome. So each gene or allele, and remember, allele is a version of a gene, right? So like whether you have blue eyes or brown eyes, you have the blue eye allele or the brown eye allele of the eye color gene. Okay, let's start over. So each gene or allele is found at a specific location on a chromosome. This is called its locus. Now, the plural of locus is loci. Yep, see? Loci. So when I say loci, it means multiple locations. You can think of a locus as an address. It's the address where that gene resides. So genes have the same locus on both members of a chromosome pair. Okay, so chromosome pairs, right? You get one copy of a gene from your mom and one copy of a gene from your dad. So if this is the chromosome from mom and this is the chromosome from dad, right? The gene for height locates the exact, or is located at the exact same address on both chromosomes. This is going to become important when we start talking about meiosis and crossing over and genetic variation. But for now, what you need to remember is that genes have an address on chromosomes. That address is called a loci. And that address remains the same. So if I collected, 1 million genes from people in Tampa, right? Or a million chromosome number ones from all the people in Tampa, the location of the gene for height would be in the same place on all of them. It's a roadmap. And that makes sense. You've heard me use the analogy that your DNA is really the instruction booklet, right, to build you. All that means is that everybody's instruction booklet has the same table of contents. The pages go in the same order for everyone. Each gene has its specific location on its specific chromosome, and that's the same for everybody. Understanding number 3. The various specific forms of a gene are alleles. So variations of a single gene are called alleles. There are lots of different alleles for genes that determine phenotypic traits that we see. Things like hair color, blood type, skin tone, ear shape. If we're talking about plants, those things can be flower color, seed color, or height. Now, if we're talking about hair color, it's controlled by one trait, but there's lots of different versions of hair color. Therefore, there are lots of different alleles for the gene of hair color. Understanding number 4. Alleles differ from each other by only one or a few base pairs. Although alleles are able to cause variations of a trait, their DNA sequences are often only different by one or a few bases. Small changes in DNA can cause a change in the amino acid sequence of a protein, and this change can have subtle or sometimes drastic effects on the characteristic. Now here you see 3 sequences of DNA. What you see at the end are 3 different colors: yellow, red, and blue. If we imagine that these phenotypes, or these colors, are they would be the different colors of feathers on a bird. So say we have a population of birds, and those birds can be either yellow, or red, or blue. All the same bird, same species, right? But they can be 3 different colors. Kind of like humans have a ton of different skin tones, right? Well, this color is only controlled by one gene. So these sequences are 3 different versions of the same gene. So the gene would be the gene for color, right? And this is allele 1, which would be yellow. Allele 2 would give you red. Allele 3 would give you blue. And if you notice, there's not many differences. There's like 1 right there. And another one right here, right? So alleles are very, very similar. They're different versions of a gene that differ by only a couple of base pairs. But those couple of base pairs make a pretty significant difference in the trait that you see expressed. Understanding number 5. New alleles are formed by mutation. Mutations are changes in DNA that can occur randomly or as the result of external factors. If mutations affect the structure or characteristic of a protein, they can result in a new allele. And when you did the transcription and translation lab, the mice gene expression lab, this is exactly what you were doing. You were looking for changes in the DNA structure that caused differences in the amino acids, which caused a difference in the phenotypic trait that you saw. So here we have our, let's see, it's got a U, so this must be RNA, right? Here's our RNA. Nope, this is, RNA, right? And then this is our amino acid, right? And here we have an insertion where instead of, let's see, same, right? CGA, that's our codon for arginine. It's the same down here. UUA, that's a codon for leucine. Same down here. Okay, but now there's a change, right? Instead of UAC, we inserted an A. And instead of tyrosine, now we get leucine. Now that A shoved that C over, so now the next amino acid is CGG, which gives us arginine instead of glycine. This is a frame shift mutation because it shifts everything after. Now that means every single amino acid after this point in that protein is going to be changed. Another example is a deletion, where we delete that C, which turns that UAC into a UAG, which stops. And this is a nonsense mutation. This should be a review from the lab that you did, right? If you're still not sure about that, that's okay. Go back and look at the PowerPoint that goes along with the HHMI lab. And we're going to talk about more examples too. Oh, here we have a substitution, right? We changed that GAT into a TGC. Oh, because here's the original up here. Ha 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 ha. So TGC, right? And then we changed it. Okay. Now let's put some of this information together. Application number 1. The causes of sickle cell anemia, including a base substitution mutation, the mRNA transcribed from it, and a change to the primary amino acid sequence of a protein, and that's a missense mutation. So sickle cell anemia is an inherited disease. A mutation in the hemoglobin gene causes the hemoglobin protein to change its shape, which gives cells a sickle shape. Because of this, the cells are not able to carry as much oxygen as they would normally. That sickle shape also can result in clogged capillaries. And this disease is pretty devastating. So symptoms include fatigue because you can't deliver as much oxygen to your organs and muscles as necessary, pain in the joints from when those arteries and blood vessels are clogged, and a breakdown of red blood cells. The change in the hemoglobin results from a single base substitution mutation. You're changing one T. You change that T to an A, right? That causes a change from glutamic acid to valine. One single letter in over 30,000 genes can result in a disease like sickle cell anemia. It's just kind of incredible to think about. That's like if you wrote a 1,000-page paper and you made a spelling error of one letter and one word in just the right place. And that would change everything about the entire paper. It's amazing. So let's see exactly how this works. Now, you guys probably remember LabXchange. I had you sign up for a LabXchange account earlier in the year. Now, LabXchange doesn't let me embed their videos, so you're going to have to go to your LabXchange account to watch this video. And this video is going to help satisfy the objectives that are listed under A1. By the time you're done watching the video, you should be able to state the cause of sickle cell anemia, including the differences in the HB alleles, that's the gene that controls hemoglobin, HB. You should be able to state the difference in the amino acid sequences in the transcription of normal and mutated HB mRNA. And you should be able to outline the consequences of the HB mutation on the impacted individual. So you should be able to click this link if you're going to download the PowerPoint, or you can just go to LabXchange. Oh, sorry, that's my Microsoft Teams. Okay, so you should be able to go to LabXchange, make sure you sign in. I'm already signed in, I think, right? Yep, I'm signed in. And you're going to go to your class, juniors. And so for you guys, these are videos we watched previously. Here we go. Here it is. Sickle cell anemia, functional validation of genetic mutations in human disease. So I'll play it right now, and you can go back to the link yourself and watch it as well. Except for that I can't hit the play button because my video controls are in the way.
Sickle cell anemia, functional validation of genetic mutations and human disease. Tiny variations in the human genome can be significant when they impact protein function. A well-known example occurs in sickle cell anemia, a hereditary blood disorder that dramatically affects the shape of red blood cells and hinders oxygen transport throughout the body. Normally, red blood cells are round and flexible, traveling easily through the narrow blood vessels. With sickle cell anemia, hemoglobin molecules form abnormally and hinder blood flow and oxygen transport. Hemoglobin is a molecule in red blood cells that carries oxygen to cells throughout the body. People with sickle cell anemia have a single base pair mutation in the hemoglobin gene's coding region, where a thymine molecule is replaced by an adenine molecule. This in turn causes the hydrophilic amino acid glutamate to be replaced by the hydrophobic amino acid valine. Consequently, the hemoglobin structure is altered and the red blood cell folds into an abnormal, rigid sickle shape that can no longer carry oxygen as efficiently, causing pain and organ damage and resulting in a risk of various life-threatening complications that can shorten patients' lives by 30 years. Although there are many single base pair mutations, also called single nucleotide polymorphisms, or SNPs, throughout the human genome, only some SNPs, like the thymine to adenine substitution in hemoglobin, have functional implications. The identification of SNPs that have functional effects is an important area of research, as this will provide further insight into biological mechanisms of disease susceptibility and resistance, as well as human genetic diversity as a whole. There are many different methods used to identify SNPs with potential functional implications. For example, computational genome-wide association studies use supercomputers to compare the genotypes of patients with the disease to those of a disease-free control group in order to identify SNPs more commonly associated with one of the two groups. This type of study is called a case control model because it compares a group of cases with the disease to a group of controls without the disease. Such studies have revealed numerous associations between disease phenotypes and candidate genetic mutations that may confer resistance or susceptibility to the studied disease. After the candidate SNPs are identified, their functional effects must be validated experimentally. To demonstrate what this entails, we will consider the example of the hemoglobin mutation. To examine the effects of the mutation, the non-mutated and mutated versions of the hemoglobin gene will be cloned and inserted into human cells that are afterwards cultured in the laboratory. As the cells grow, they will produce hemoglobin. Comparing the hemoglobin produced by the cells with the non-mutated control gene to that produced by the cells with the mutated gene will show us that the SNP impacts the hemoglobin structure. This kind of functional analysis helps illuminate the biological effects of genetic mutations and provides further insight into the genetic basis of disease. In the case of sickle cell anemia, functional analysis allowed us to determine that the genetic mutation alters the structure of the encoded protein. However, other mutations may be located in non-coding regions of the genome, and functional analysis can show that these mutations have different biological effects, such as altering the amount of protein in the cell rather than the structure of the protein itself. In this way, understanding the biological impact of a disease-associated mutation can provide insight into the underlying physiological mechanism of the disease. Sickle cell anemia, functional validation